You know, when you stop and think about it, the sheer variety of life on our planet is just staggering. I mean, from the teeniest little bacterium to the biggest blue whale, there are millions upon millions of species. So, how in the world do you even begin to make sense of all that? Well, today, we're diving into one of the biggest organizational puzzles in all of science: how we built a filing system for every living thing on Earth.
Okay, so let's really get into it. Picture this. You're handed a library with millions of books, but none of them are sorted. No genres, no author names, nothing. That's pretty much the challenge biologists were facing. How do you create a system that not only sorts everything we see today, but also, makes sense of how all this life is connected? It's a huge puzzle, right?
Humans had been trying to organize life for, well, forever. For our ancient ancestors, this wasn't some abstract scientific quest, it was about survival. You had to know what you could eat, what could be used as medicine, and most importantly, what was going to try and eat you. Classification was practical, it was essential. So our first real documented attempt goes way back to Aristotle in the 4th century BCE. His method was pretty simple. He just used what he could see. He sorted plants by their size. Are you a tree, a shrub, or an herb? And animals by whether or not they had red blood. Now, let's fast forward a couple of thousand years to the 18th century. That's when Carl Linnaeus came along and created the formal two-kingdom system that honestly dominated biology for a really long time. But as you can probably guess from that last point on the timeline, by the 20th century, scientists started realizing this system has some serious problems. And this was it, Linnaeus' system. It was beautifully simple. You had Kingdom Plantae on one side for all the plants and Kingdom Animalia on the other for all the animals. Basically, every living thing got stuffed into one of these two filing cabinets. And for a while, hey, it worked pretty well. It was easy to understand, and it covered most of the big familiar organisms.
But here's where things get messy. As our tools got better, especially with the invention of the microscope, we started discovering this whole world of bizarre organisms that just didn't fit. You couldn't neatly file them under plant or animal. The filing system was starting to burst at the seams.
So, what exactly went wrong? Well, for starters, the system completely ignored some fundamental biological truths. It didn't separate simple cells, prokaryotes, from complex ones, eukaryotes. It just threw single-celled life in with massive multicellular creatures. And, maybe the craziest part, it put things that make their own food, like algae, in the same big bucket as things that don't, like fungi. The whole thing was just too simple. Okay, let's look at our first problem file, a mushroom. I mean, it's got a cell wall, so it's kind of plant-like, right? But it doesn't do photosynthesis at all. It survives by absorbing nutrients from dead stuff. So, is it a plant? Nope. An animal? Definitely not. It's a classic case of a file that just doesn't fit. And what about this little guy? An amoeba. It scoots around and eats things, which sounds pretty animal-like, but it's just one single cell. Does it really make sense to put this in the same kingdom as a lion or a whale? It felt wrong. Another problem file. And then you've got bacteria. I mean, these things are everywhere, but their cells are incredibly simple. They're prokaryotic. Lumping them in with complex plants just because some of them have a cell wall, that's like filing a one-page sticky note in the same drawer as a 1,000-page encyclopedia. It just makes no sense.
So by the middle of the 20th century, it was pretty clear the old system was broken beyond repair. Biology was desperately in need of a brand new filing system. And the person who stepped up and delivered that new blueprint was an American biologist named R.H. Whitaker.
And there it is. In 1969, Whitaker proposed a completely revolutionary new system. And this wasn't just a small update, it was a total rethinking of the rules of the game, blowing up the two simple kingdoms into 5 much more logical ones.
Now, here's the really brilliant part. Whitaker didn't just add more folders to the filing cabinet. He invented a whole new set of filing rules. He argued we needed to look at much more fundamental stuff. How complex is the cell? How is the organism's body put together? And, this was the real game changer, how does it get its food? And this chart just lays out his elegant system perfectly. Check out that top row, cell type. Right away, he creates a whole new kingdom, Monera, just for the simple prokaryotes. Problem solved. Then, for all the complex eukaryotes, he looks at body structure. Are you just a single cell? Okay, you go in Protista. Are you multicellular? All right, now we look at the mode of nutrition. Do you make your own food? You're in Plantae. Do you absorb it from your surroundings? You're a fungus. Do you eat it? Welcome to Animalia. It's just so logical, and it solves all those old problems.
And boom, there you have it, the 5 kingdoms, each one defined by a crystal clear set of rules. We've got Monera, Protista, Fungi, Plantae, and Animalia. This completely changed the game and became the new standard for how we see the tree of life. So how did this new system give us a better picture of life? Let's take a quick tour through some of these new kingdoms and really focus on how they finally gave a home to all those misfits from the old model.
First up, Kingdom Monera. This became the exclusive club for all prokaryotes. All those bacteria that were so awkwardly shoved into the plant kingdom, they finally had their own place, one that recognized their unique and much simpler cellular design.
Next up, Kingdom Protista. Now this is a really fascinating one. You can think of it as the miscellaneous drawer for any eukaryote that isn't a plant, an animal, or a fungus. It brought together all those single-celled organisms like amoeba and euglena and really highlighted their role as the ancestors to the other, more complex kingdoms. And finally, our original problem file gets its day in the sun. Fungi were finally recognized for what they are, a totally unique group of heterotrophs that absorb their food. Plus, we realize their cell walls are made of something called chitin, not cellulose like plants. They absolutely deserve their own kingdom. But here's the thing about science, right? The story is never really over. A textbook is never truly finished. Just like Linnaeus' system had to make way for Whittaker's, our own understanding just keeps on changing as we find new evidence. In fact, the story's already moved on. These days, a lot of scientists use what's called a 3-domain system, which is kind of like another layer on top of the kingdoms. And based on some deep genetic discoveries, it actually splits Whittaker's kingdom Monera into 2 completely separate groups, the archaea and the bacteria. It's just the next step in this great filing project. So that leaves us with one last big question. This tree of life we've been talking about isn't some stone tablet. It's more like a living document, one we're constantly editing and redrawing. As we discover new forms of life and invent new technologies to study them, you have to wonder, what new branches are we going to add? And what will our picture of life on Earth look like next?